Hello, Biology 230. This is Dr. Chastity Bradford at Tuskegee University, and I will be presenting Chapter 11, Concept 11.3. As we continue to discuss signal transduction and signaling, transduction, of course, is the second step in this three-phase process. Okay? And the last step, of course, is a response. What does transduction have to deal with? There are a number of signaling cascades. What are these cascades? They're molecular interactions that relay signals from the receptor to target molecules in the cell. Now, signal transduction involves a number of steps, and this is how the cell, with all these pathways, this is how the cell amplifies its signal. So you have your ligand that binds to its receptor on the outside of the cell, and then this signal is then transduced internally in that cell, intracellularly. Okay, so you have a few molecules, they produce some large cellular response. So if you recall, there was this show a long time ago called Pimp My Ride, and what the director of the show would do, what they would do is, it was a part of MTV, and they would come and they would put all of these amplifiers and speakers and just, soup up someone's automobile that had been, some clocker that they had. And so I want you to think about these amplifiers and think about the fact that this is exactly what the cell does in terms of utilizing its transduction mechanism and signaling cascades and these transduction pathways inside. There are enzyme cascades inside the cell that amplify the cell's response. So there is a ligand, it binds to its receptor, and then the cell responds in a way by which signals are transduced. Okay? And these enzymatic cascades amplify the cell's response to that signal. And at each step, the number of activated products is greater, of course, than the preceding step. Now, what else do we know about these signal transduction pathways? Just like a relay race, the molecules themselves, they relay a signal from the receptor to the response. And most of these molecules involved in relaying this signal are mostly proteins. So just like falling dominoes, the receptor will activate another protein, which activates another, and that activates another until we have a response. How does this actually take place? Protein phosphorylation is involved, and this is the common mode of regulation in cells, and it is a major mechanism of signal transduction. Understand protein phosphorylation, okay? Because in many pathways, the signal is transmitted by a cascade of protein phosphorylations. Now, who's responsible for these protein phosphorylations? Protein kinases. And by now you've heard about kinases. Okay? Now, protein kinases transfer phosphates, phosphates from ATP to some protein. And this process is called phosphorylation. And if you recall, in our previous lectures, previous chapters, we talked about substrate level phosphorylation and oxidative phosphorylation. Both of them involve transfers of phosphate groups. Okay? In this case, we're transferring them from ATP to a protein. And this is the major mechanism of signal transduction. Okay? This is how we regulate protein activity. And most of the protein kinases, they're going to act on substrate proteins. Okay. Now, as you recall from the previous slide, kinases transfer phosphate groups from ATP to protein. So in this instance, we have a signaling molecule or some ligand binds to its receptor. Oftentimes, this receptor changes its conformation or changes its shape. And then that signal, that external signal, has to then be relayed internally until there is some cellular response. So what you see here is the receptor is activated. This signal is then transduced. Okay, so this is a relay molecule. We have an inactive protein kinase, and it becomes an active protein kinase. How does it? It becomes an active protein kinase. And when it's activated, what do we say protein kinases do? They transfer phosphate groups from ATP to some protein. So this active protein kinase will transfer a phosphate group from this ATP to some protein, and then in this instance it's transferring it to the active protein kinase 2. So now this protein kinase is now activated, okay? And then this signal is then transduced to active protein kinase 3 till we get to the active protein that then has its cellular response or creates a cellular response. So how is active protein kinase 2 activated? It transfers a phosphate group. Well, active protein kinase 2, of course, was activated from active protein kinase 1. Now, active protein kinase 3 is activated because of the transfer of the protein of the phosphate group from active protein kinase 2 to active protein kinase 3. And then active protein kinase 3 transferred the phosphate from ATP to active protein here, the active protein here. And this activated protein then creates some cellular response. Now, what I want you to remember is that most of this phosphorylation that you see here, that's what this is. This is a phosphorylation cascade. Now, most of it occurs at either a serine or threonine amino acid residue, okay? Now, if it's a tyrosine kinase, there will be, of course, a tyrosine that's going to be phosphorylated, and you'll see that later on, okay? So know that many of the relay molecules, okay, these are relay molecules, starting here from this one that's activated. Many of these relay molecules are protein kinases, and they act on other protein kinases to create this phosphorylation cascade. Now, each one of these protein phosphorylation leads to a conformational change because of the interaction between this newly added phosphate group and the charged or polar amino acid on the protein. So every time this occurs, there is some conformational change, okay? And that makes sense because remember, they're proteins. And so when this signaling molecule binds to this protein, there is some conformational change or shape. The shape changes, okay? Now, most of the time, phosphorylation leads to activation, okay? Not inactivation. It often leads to activation. But you notice here that they're inactive proteins. So this inactive protein kinase 2, there's an inactive protein kinase 3, an inactive protein here. How does that happen? And notice that the shapes are different, okay? Same protein, but for some reason it's in an inactive state here. How does that happen? We have to have some mechanism by which we regulate this phosphorylation. And it is controlled by dephosphorylation, the removal of phosphate groups. So how are they removed? Protein phosphatases, again, enzymes, they remove phosphates from proteins, and this is called dephosphorylation. So the phosphorylation, dephosphorylation system, it acts as a molecular switch, turning activities on or off or up and down, okay? So the regulation can be completely off, or we can decrease the signaling or increase the signaling. So these are what you see PP here in purple. These are phosphatases. So they're responsible for removing these phosphate groups. So that's another level of regulation. So you remove this phosphate group and inactivate protein kinase 2. The removal of this phosphate group here will inactivate protein kinase 3. The removal of the phosphate from this active protein creates an inactive protein here. And so you would not have a cellular response after this phosphate group is removed, and that's how we regulate it. So once that cellular response has occurred, these phosphatases come in and they inactivate the proteins, or they inactivate these protein kinases, okay? And this is the level of regulation that exists for our cells. And what happens is these phosphatases are then making these protein kinases available for reuse, so that when that next signaling molecule comes in, they're ready to be activated. And it enables the cell to respond again. Okay? At any given moment, the activity of protein regulated by phosphorylation is going to depend on the balance between active kinase molecules and active phosphatase molecules, okay? So there's going to be a balance between the two. Now, what is going to happen if this signal is not here? If you don't have this extracellular signal here, what do you think happens? You're going to have a more protein phosphatases will predominate. If there's no signal here, you'll have more protein phosphatases predominating, which makes sense, right? Because now this signaling pathway, this cellular response will be shut down because there is no signal there, okay? Now, there is another component to this, and they are called second messengers. And so that's where we're moving into this next phase that we're called, please don't shoot the messenger. So in signal transduction, there's second messengers. Now, before we go into the second messengers, this is something that a number of people drink, college students as well. But what is really keeping you alert? I drink coffee. What's in it that keeps me alert in these Monster energy drinks? What you see in, excuse me, there's ginseng and there's also caffeine. So how does caffeine really work? I'm going to illustrate it to you. So as we mentioned before, adenylyl cyclase is responsible for converting ATP to cyclic AMP. Now, cyclic AMP, of course, is our second messenger. So what coffee actually does is it increases your cyclic AMP. And it does this by preventing the conversion of cyclic AMP to AMP. So ATP is converted to cyclic AMP using adenylyl cyclase. Cyclic AMP can then be broken down to AMP, and coffee, or caffeine, shall I say, it prevents this conversion from cyclic AMP to AMP. So therefore, it's always active, if you're chronically drinking it. Now, what can be the cellular response then of increased cyclic AMP? So in the brain, it can lead to alertness. In blood vessels, it can cause constriction, which will increase your heart rate. So your heart starts beating fast and you're alert, bright-eyed and bushy-tailed and ready for whatever the day may bring. Or in midday, you drink your Monster or your caffeine and you realize that you're alert and you feel your heart rate increasing if you have a lot of it. And so this also illustrates the fact that the same signal can have a different response depending on the cell. So in this instance, we've increased cyclic AMP levels with caffeine, but in the brain it caused alertness, but in the blood vessels it caused vasoconstriction, which increased heart rate. So this is another pictorial diagram demonstrating what happens, how we get cyclic AMP, and then how caffeine increases AMP levels, cyclic AMP levels. Okay, so this is ATP. So remember, we have triphosphate here. Now, adenylyl cyclase is responsible for converting ATP to the cyclic form called cyclic AMP. Okay, now cyclic AMP can then be broken down to AMP by a cyclic AMP phosphodiesterase. Okay, now if this does not happen, if this cyclic AMP is not broken down to AMP, then of course you'll just have a lot of cyclic AMP around, and the signal that occurs in response to that in the brain is alertness, and in the heart is an increase in heart rate, or your blood vessels will constrict. So once again, we have ATP being converted to cyclic AMP using adenylyl cyclase, okay? And then cyclic AMP can then be broken down to cyclic AMP. Cyclic AMP can then be broken down to AMP via a phosphodiesterase. So caffeine is a phosphodiesterase inhibitor. So it prevents this conversion from cyclic AMP to AMP. Therefore, your cyclic AMP levels remain high. You remain alert. Your heart rate, if you drink large amounts of this caffeine, will increase. Cyclic AMP is one of the second messengers. So now we'll move into first and second messengers. So there are different types of messengers. The ligand, or that extracellular signal molecule, it binds to the receptor, and that's the first message, or the first messenger. Now the second messengers, they're small, non-protein water-soluble molecules. They could be ions as well, and they spread throughout a cell by diffusion. The fact that we said they're small, water-soluble, should let you know that they can easily diffuse into the cell, but they diffuse throughout the cell by diffusion. Okay. Second messengers participate in pathways initiated by G-protein coupled receptors and receptor tyrosine kinases. And if you recall earlier, I mentioned to you the fact that in G-protein coupled receptors, there's serine and threonine residues are often within your G-protein coupled receptors, but the receptor tyrosine kinases, they will have tyrosines. That just makes sense, right? Okay, so that is something else that I would like for you to remember, that most of the phosphorylation is going to occur at serine or threonine amino acids in these G-protein coupled receptors, but in receptor tyrosine kinase, it's going to be tyrosine residues. Now, cyclic AMP and calcium are the common second messengers that are used. Know that cyclic AMP and calcium are common second messengers. What do we know about them? They can rapidly diffuse, so cyclic AMP, calcium we'll talk about in a minute, but they rapidly diffuse throughout the cell. Now, second messengers participate in pathways initiated by both G-protein like link receptors and tyrosine kinase receptors. And this is a G-protein coupled receptor, which means this is the receptor and it's coupled to a G-protein. Very simple, right? And this is the first messenger, and the second messenger is cyclic AMP. Cyclic AMP is a second messenger. It's one of the most widely used second messengers. Adenyl cyclase, or adenyl. You might hear people say adenyl, adenylyl, but it's adenyl, adenylyl cyclase, adenyl cyclase. Okay, so cyclic AMP is a second messenger. Cyclic AMP is one of the most commonly used second messengers. And then adenyl cyclase is an enzyme in the plasma membrane. It converts ATP to cyclic AMP in response to that extracellular signal. What's interesting, and you'll see, is that these extracellular signals, all they do is initiate this whole process. They usually don't come into the cell. They just send a signal. They sit on the extracellular side of this membrane and send a signal into the cell, and then that cell somehow responds. It is very amazing. Now, this is another view of the first messenger and the second messenger, and we're just reiterating the fact that signaling molecules, they trigger the cyclic AMP formation. You have your G-protein coupled receptor. You have your G-protein here, and you see GTP. This G-protein is activated when you see GTP being active. Adenyl cyclase, so the G-protein will diffuse across the membrane, bind to adenyl cyclase, and then it's then active, and then you have your second messenger that then forms. When this signal occurs, it causes or triggers the formation of cyclic AMP. Okay, now what does cyclic AMP then do? It's a second messenger, but it then goes on and activates protein kinase A, and it can phosphorylate, which can then phosphorylate various other proteins. Now, you may be asking, why cyclic AMP? What's so amazing about it? Now, the normally the cellular concentration of, am I highlighting? Yes, the cellular concentration of cyclic AMP is one that we can change really fast. Within seconds, you can boost it 20-fold. Okay, it has a short half-life, or and the phosphodiesterase is a way that then is converted to AMP. So just like ATP goes to ADP, cyclic AMP is then converted back to AMP. So its active form, if you will, in terms of this signaling cascade is cyclic AMP. Now, calcium is another second messenger. So far, we have established that transduction pathways, they amplify a signal. We have established that there are first messengers and second messengers, and the second messenger earlier that we talked about was cyclic AMP. Now, there is another one that's widely used, and it's calcium. Okay, so cyclic AMP and calcium are both second messengers that are widely used. Why do you think calcium is used? Its cytosol concentration is lower than its extracellular concentration, and because of that, the cell can then use it as a second messenger. So what you're seeing here, if this is our cell, this is our plasma membrane here in orange, that in the cytosol, there's a low calcium concentration, and out here in the extracellular fluid is higher. And so on these organelles, on the endoplasmic reticulum, there's a calcium pump. You see how there's ATP there, so this must be actively transported. Here in the mitochondria, there is a calcium pump here as well, and you see that on the plasma membrane, there's a calcium pump. Okay? And so once again, if there is a pump, then it's going against this gradient. So in this case, calcium then is going to be pumped out of the cell, okay, as opposed to going with its gradient, which is higher outside. And so the cell uses this as a second messenger. Now, what happens? A signal is relayed by the signal transduction pathway, and this triggers an increase in calcium in the cytosol. Now, there are a number of signaling molecules that are involved in this signal transduction pathway that leads to the release of calcium, IP3 and DAG. IP3 is inositol triphosphate, and diacylglycerol is DAG. Okay, now they're also second messengers. So what you see here is the first messenger, which is the ligand. It binds to the G protein coupled receptor, and you see that this G protein is activated, and when it activates, it diffuses across this membrane, and it activates phospholipase C. And then when that happens, PIP2, you can notice here, becomes split into diacylglycerol and IP3. And these are second messengers. You notice that diacylglycerol stays within this component of PIP2, stays within the plasma membrane, but the other component of PIP2 is released into the cytosol. Okay, and this then functions as a second messenger. How does it do that? Now IP3 then can bind to the calcium pump on the endoplasmic reticulum, and then calcium then can be exported from the ER into the cytoplasm, and that can change that cytoplasmic concentration of calcium that normally is low. Now it's higher, and this is a signal because it's not the normal thing that the cell sees. Normally in the cytosol, the calcium levels are low, and so the second messenger, IP3, once again, it'll bind to the IP3 gated calcium channel, okay? And then when it binds to that channel, remember, conformational changes occur, and it occurs in a way that opens up this channel, and then calcium can be released into the cytosol. Now, when that happens, other proteins can be activated, and then depending on what proteins are activated, that dictates what cellular response is then comes as a result of this. Now,